So hello all and welcome to this expert panel session on Rare Diseases Reimagined. My name's Nabiha Sachdina. I'm Vice President of Health Programmes here at Oxford Nanopore. And in this session, we've got 5 distinguished speakers. Each of them in turn will be asked to come up to give a 5-minute introduction to their area of work, and then we'll follow that with a panel discussion. And the aim here is really to explore some of the future challenges and opportunities to fully realising the power of genomic sequencing to human health. We will be taking questions for the panel from the floor and via the app. If you're using the app, please remember to use the thumb icon to upvote your favourite questions. I encourage you to ask questions to the whole panel where possible, but if you do have a question for a specific individual, please just refer to them by name in the question. So now I'm delighted to welcome our first panel member, Professor Joris Vermeesch. Joris is a Professor of Molecular Cytogenetics and Genome Research and a staff member at the Centre for Human Genetics Leuven, Belgium, and he's also Scientific Director of the Genomic Core Facility at KU Leuven. He is President-elect of the Preimplantation Genetics Diagnosis International Society and board member of the International Society of Prenatal Diagnosis. His laboratory develops and translates novel genomics methods in preimplantation, prenatal and postnatal diagnosis, and more recently, cancer screening. They've pioneered array CGH, single-cell arrays, massive parallel short and long-read sequencing, and cell-free DNA analysis. So welcome, Joris.
Hello, it's my pleasure to be here and to be invited to this crowd, and especially to meet quite a number of people I knew from literature, but get to know them a little better, so it's my pleasure. I was asked to give in 5 minutes my view, or maybe give a little bit of palette of what we're doing with ONT and long-read sequencing at the moment. As you heard from my bio, I have a history in constitutional cytogenetics, which entails 3 areas. It's reproductive genetics, prenatal genetics, and postnatal rare disease genetics, let's say, and trying to understand the causes of rare diseases. What we heard already a lot on this meeting was about how long-read will streamline probably the rare disease genetic diagnostic field. We will integrate, or we are in the process of integrating different separate diagnostic analysis pipelines into one long-read platform, and I think ONT is amazing to do that. We heard Danny Miller speak this morning. Also, in a later session, we heard how we can integrate the methylation data and the information derived from the methylation into the analysis of rare diseases. I've been exploring 2 other areas for long-read sequencing. As I mentioned, I'm working in reproductive genetics diagnosis. I have a history of pushing technologies to enable single-cell haplotyping and genotyping. We used to do that with arrays, SNP arrays. Then we moved on to targeted sequencing, short-read sequencing, and now we're exploring how the long-read sequencing may actually also revolutionize that field of preimplantation genetic diagnosis. We should realize that once we have made a diagnosis postnatally, parents are always shaky and wondering, how about my next child? And then so the only approach to solve that is to through PGT, and that's where we can make a difference as well. And if you're interested, you can ask questions and I'll discuss that a little bit more with you. And then last but not least is in the prenatal field, where obviously, especially the fast track, the potential of ONT rapid sequencing may also change that field. Currently, we usually following non-invasive or methods of detection. This can be ultrasound. We detect abnormalities in the fetus. Usually we do an invasive follow-up, and that the analysis is done. Nowadays we do whole exome, whole genome sequencing. It typically takes 5 to 10 days before we report an answer. But with the fast-track ONT, I think we may change that to reporting results in 1 day. And I think that's really an amazing future potential. And these are the areas that I'm actually exploring. Yes, that's probably my 5 minutes token. Thank you.
So thanks so much to you, Joris. And before I introduce our next speaker, just a reminder to keep any questions going in on the app, please. And now it's my great pleasure to introduce our next panel member, Nick Sadden. Nick is Chief Executive of Optum Genomics and SVP at United Health Group in the US. He previously served as Special Advisor for Health and Life Sciences for the UK Prime Minister David Cameron, where he helped to develop the sustainability strategy for the UK's National Health Service and part of the programme to sequence 100,000 genomes. Nick is a visiting professor at the Imperial College of London Institute of Global Health Innovation and also sits on the Cambridge Life Sciences Advisory Council, and he was awarded an MBE in 2016. Welcome, Nick.
Thanks very much indeed, and thanks so much for having me here. You might wonder how a Brit ended up in the Midwest in Minneapolis. Britain voted for Brexit when I worked for Cameron, and then I did my own Brexit and decided to get out and do something totally different. As a consequence of having been involved in Genomics England and then doing what I do now, it's my fortune or misfortune to always be surrounded by people that are more scientifically expert than me, and this is one of those moments that's a little bit scary. Looking at the collective audience, I thought before I go, just keep an eye on that timer. I want to spend a couple of minutes just explaining United Health Group, because the context of the organisation will help explain why we care about genetics and genomics as much as we do. So United Health Group is massive. It's a $400 billion revenue company in the US, and half of it is a payer, and half of it is a services and care delivery organization. So we have a lot of data, which we use, run analytics over for a whole range of different purposes. We have a large care delivery, multi-specialty and primary care group capability with 90,000 physicians. And we have a large pharmacy care organization as well. And for us, we see, we believe that Siddhartha Mukherjee was right, that if the 20th century was the century of the atom and the byte, then the 21st century will be the century of the gene. And when I super quickly described the parameters of our organization, but we see the potential to move into a world where you have a precision formulary for medicine so that you can reduce adverse drug events and optimize therapies to ensure that the massive potential to improve the quality of care and reduce cost in the way that we deploy therapeutics across our organization and the healthcare system more broadly. For our physicians, the ability to predict and prevent, intercept and prevent disease carries great hope across, whether that is in the primary specialty, hyperacute space. By the way, I am conscious that I'm speaking unapologetically as a sort of health system person in the conversation, but I could go on monogenic and polygenic disease, polygenic, the ability to kind of get after large population groups and improve the quality of care. Again, very promising. There are a number of very big challenges for healthcare organizations with the space, with the market, and it's worth saying something about the market conditions in America, at least. You and Ashley at Stanford makes the point that if the cost of a Ferrari had come down by the same cost as the whole genome over the last 25 years, you'd be able to go out and buy a Ferrari for 40 cents. So the cost compression is absolutely incredible, and of course, therefore, not part of the reason why genomics has not penetrated more broadly in healthcare. One of the biggest challenges is that in America, it is a largely unregulated space. So to give you a sense of what that means that a test developer can launch a test to market without actually having the FDA review the quality and validity claims. Not all do, but many do. And so we have 175,000 molecular diagnostics on the market. And one of the challenges for that is that you, of course, have variability in quality and variability in cost. And if you're a provider, trying to know which test to order from which lab is super difficult. If you're a payer, knowing which test to cover and reimburse is super difficult. and you get a lot of abrasion in the system as a result of that. If you're a test developer and a manufacturer or lab, you're also really frustrated because you're struggling to get adoption. Or you're an investor, you're really frustrated because you're struggling to get a return on your investment. So we can talk about what we're doing more in the discussion, but we're doing a number of things now because we do believe in the future and we believe that the challenge is tractable. We're working very hard to bring in. We have a huge amount of clinical and claims data. We're looking very hard at how we can use genomic data, the raw data files, but also test results from testing on our populations to improve our understanding of what's going, not just biomarker discovery, but also what's actually happening with utilization patterns or adherence to guideline recommended testing in the US. So we've got a data business. We have a test management business, which is focused on how we better manage the appropriateness of different test ordering and how the medical necessity and all sorts of things like that are being documented or not documented. And we have a consulting and research business that works with labs and test developers to help in the path towards coverage and reimbursement and adoption. And this is really about providing technical assessments on the published evidence for different intended uses. It's about market access consulting so that you can understand exactly what some of the puts and takes are about bringing a test to market, what the payers are really trying to understand. And we also do both retrospective and prospective evidence generation with the people that we're working with, including Oxford Nanopore, to try and, again, accelerate that path to market. So we can say more about some of the other programs that we're working on or go into any detail on that. But I'm the sort of the token generalist on the discussion panel. So if you've got questions about how we actually integrate this extraordinary science into healthcare, then that's my area of special interest. Thank you.
Thank you, Nick. Thank you, Nick. And I'm sure there are going to be many such questions which will be coming your way and the panel's way during the panel discussion. But for now, I'm delighted to welcome our next panel member, Danny Miller. As you recall from his earlier plenary presentation, Danny is the Assistant Professor in the Department of Pediatrics, Division of Genetic Medicine, and the Department of Laboratory Medicine and Pathology at the University of Washington. And because that isn't enough, he's also an attending physician at Seattle Children's Hospital. Welcome, Danny.
Thank you. And it's a little odd to get up and speak after speaking earlier this morning. So I'll be, I think, very brief because I told most of you what I think the future of rare disease diagnostics is or why it's long-read sequencing. I just want to give you a couple of examples kind of from my clinical experience, why I think long reads is the path forward. And for me, it's a typical day in clinic where I might see 6 patients. Let's say 3 of them are new patients, but 3 of them are return patients. And for those return patients, there's probably 1 or 2 who have gone through a variety of tests. They've had an array, they've had a panel, they've had an exome, and those have all been negative. And we sit down and we essentially review, what has happened over the last year since I saw you last, and then think about what's the next test that we could send in order to, try to understand what's going on. In some cases, those are kids who, parents really want to know for recurrence risk, right? In other cases, those are kiddos who their parents want to know, is there a treatment? Is there precision therapy? Are they eligible for an ASO, an antisense oligonucleotide, which more and more families are actually coming to us with this idea of, can we use this, right? So in those cases, that's really hard because I don't have a lot to offer them from a clinical perspective. The conversation kind of quickly goes to, well, what about the Undiagnosed Diseases Network in the U.S.? What about just going to my lab and getting long-read sequencing, right? But are we really tooled up to offer that at scale? We're not. So what I want is for those patients to have just gotten long reads first. We can do the complete analysis and then very quickly transition into, the research space, right? Within a week, within a day, within 5 hours, honestly, we could take that long read data, send it to a research lab, and then start asking questions about some of the variants that we find. Because, that's what those families deserve. That's what those kids deserve, right? And then alternatively for those new patients, it's just that rapid, quicker time to diagnosis. Some of them may never need to come back to a geneticist, but others may be diagnosed with something that does require long-term genetics follow-up. So in my mind, long reads is this way to simplify the entire process It drives, I think it'll lower clinical costs overall, and I'm interested in thinking about how do we quantify that, right? But also I think it drives quicker time to treatment. I'm really looking forward to the day when I walk in the NICU and we had sequenced a kid who came in the night before, and we know that they have a particular variant and it's amenable to an ASO therapy that was designed and tested a year or two ago, and we can get that drug to that kiddo within a day or two, right? That's my dream, and that's where I think long reads will get us. So I'll stop there.
Thanks so much, Danny. Thank you so much. And I'm now delighted to welcome our next panel member, Professor Emma Baffel. So Emma is Professor of Genomic Medicine at the University of Exeter, a consultant clinical geneticist at the Royal Devon University Healthcare NHS Trust, Medical Director for the South West Genomic Laboratory Hub, and Medical Lead for the NHS National Rapid Genome Sequencing Service for acutely unwell children in England. As Clinical Director for the South West Genomic Medicine Service Alliance, Emma is the responsible officer for delivery of the NHS Rare and Inherited Disease NHS Genomic Network of Excellence. And she's also co-chair of the NHS England Rare Disease Test Evaluation Working Group. So lots to offer this discussion. Welcome, Emma.
So I guess Danny said a lot of the things that I would want to say, really, in that we're both coming from the same perspective. Fundamentally, I'm a doctor, and my goal is that I'm a genetics doctor, and my goal is that we get an answer for patients in a timely manner, and that we do that in a health system that is a nationalised health system, so we can do that in a cost-effective manner. So for me, what I want from long-read sequencing is sort of the same as what Danny said, but actually I'm hoping that we can get there a bit faster in terms of my vision of how we might start to bring it into diagnostics right now. So when I spoke last year, I talked about how we in Exeter provide this service for children who are acutely unwell on intensive care, on NICU or PICU. And I gave some examples of children whose lives have been saved by that service. We aim to turn around those results in less than 10 days, ideally in 5 days from the time that the sample gets to us. And the sorts of children we see are children who have very severe epilepsy. Every time that child is seizing, they are damaging their brain. So the faster we can get that result, the better. And I totally agree with Danny. What ONT sequencing offers is an opportunity to do that at a really high pace, and you and Ashley and others have shown us that. But at the moment, what I see as the issues with long-read sequencing are we're still learning. When I look at short-read sequencing in patients that we see through that service, we can visualise that data really clearly. We've got very stable chemistry, everything's automatable, and it works in a diagnostic setting. I was really excited when I came and spoke last year, and I'm still really excited about the possibility of nanopore sequencing. But I had to sort of slightly take my really optimistic hat off and say, okay, we're not going to be doing trio sequencing with long-read sequencing. We're not going to be sequencing both parents and both child by this time this year, because we're still learning so much about what we're doing. But it's added already in our hands answers for patients in diagnostics. So what we can do with it now, what it does do, is it allows us to take some of the inequalities that we've had in healthcare. For example, when a child doesn't have one or both parents available, we can look at phasing of variants that we can't do with short-read sequencing. So we've been able to use that information already to phase variants and to provide answers for families. Structural variants are a real pain when you're looking at short-read sequencing. We've been able to provide answers for families in our sort of research to diagnostic space, because our lab is entirely integrated in Exeter where we provide that service. We've been able to do that now in real time for children who are getting testing through the acute service. And so my goal over the next year actually is for us to get that bit of the test into NHS diagnostics. And the funding that we've had from NHS England to work with all of the labs across the country and to work with providers like companies like ONT will enable us to do that. So I guess I'm standing here saying I will get long-read sequencing to NHS diagnostics within a 12-month period. It won't be me. It will be I'm the face of the Exeter Rapid Genome Sequencing Service. I'm not the person who does all the work. Andy, who is the head of our lab, Caroline Wright, who many of you will know from the DDD project, all of the scientists who work on that service, they're doing all this work right now to develop this. And we're absolutely 100% committed to getting the initial goal, diagnostic potential for long-read sequencing into NHS diagnostics, so that I can come here next year and I can say, it's an accredited test. Now we're going to do trioed genome sequencing with long-read sequencing, because I think Danny is right. For me, it is the future. It absolutely is the future. It allows us to save money. We can get rid of array CGH. We can get rid of lots of other testing that we're doing right now. With the potential to get rid of methylation testing, we can have a one-stop shop test for our patients. So it will, in the end, save the health service money. And it will, in the end, allow us to do that testing much more rapidly and much more closely to the patients. But at the moment, we need stable chemistry. We need good quality bioinformatics. We need good visualization of that data. We need to be able to automate that process so that we can do it not the way that Danny described, but the way that I reckon Danny and I both want to be doing it, which is that one-stop diagnostic test. That's me.
So Emma has very eloquently described both the ability but also the increased potential to provide answers for patients, which brings me to our next speaker, Paul. So Paul Arvidsson, it's an absolute delight to have you here. Also, I think he probably wins any competition to have the best themed t-shirts today. So Paul's an ex-theatrical lighting designer and currently a full-time carer for a daughter with an nanny rare condition. He's a writer of sci-fi featuring sentient blind guinea pigs and thrillers with a teen detective with a rare condition, a wheelchair and an attitude. Paul is also one of the poets in the carer written collection Helix of Love, produced by Brighton and Sussex Medical School and the Wellcome Trust. And he sits on the patient panel for Genomics England and is the dad's representative for Swan UK. So welcome, Paul.
Thank you very much. I feel a bit weird being called upon to be an expert in anything, really. I think it's the first time I've ever been requested to be an expert. I'm feeling kind of quite a deal of camaraderie with Nick, because we're the only people there are like doctors and professors and people who are rarefied in that nature. So I'm a dad. I'm a dad to two kids. My youngest was born in 2008, and she has a nano-rare condition. So we've got a long diagnostic journey that I can speak to later if you guys are interested in that. I'm kind of the guy with three hats, really. So there's two of them. Genomics England obviously is my first hat, without whom I would not be here. I'm on their patient panel, which for anybody that doesn't know is it's a body of volunteers. Your criteria for entry to the patient panel is that either your data or your family's data must be held by Genomics England. And our job is to ensure that there's always a face connected to that data. That there's always a voice for the numbers, for the C's, A's, G's and T's. That whilst all of this terrifyingly clever and brilliant work is going on, that it's still related to people at the end of the day. That's our job there, and to make sure that that data is safeguarded and used for the higher good. It's kind of like being a school governor, but for gene boffins. My second hat is as dad's rep for Swan UK. Swan UK, for people who don't know, I'm going to be saying this a lot. I've got three mystery hats. So my second mystery hat is for Swan UK. Swan are Syndromes Without a Name. They are a charity whose job is to represent the families and the kids who don't have a diagnosis. So if anybody was sat in here before in the previous set of discussions, Dr. Ahmed, I think, was talking about the diagnostic journey and the small part of the bar with the diagnosis people, the third of the people, and then there's the 2/3 of the people. They're all the people who live in Swan. They all arrive at Swan kind of in a desperate situation because they know something's wrong with their kiddo, but they don't know exactly what it is. And so then we go through an enormous diagnostic journey to try and find those things out. Some families like ours are really lucky and find a diagnosis, which is brilliant. Again, I can tell you a little bit more about that later on if you're interested. Other families don't, and so they're still on the same raft and it's very peculiar. And the oddest thing about Swan UK is the fact that there's masses of families there. I think the last time I knew a proper number was 2,500 families. There must be more than that there now. And they've all got staggeringly different conditions. But what is common with all those families is that we all come together in that knowledge that we know something's wrong. We've all got to go through the same diagnostic journey, and there's just so much in common. And that kind of camaraderie, I suppose, between all of those people who are going through this process and not having a diagnosis. The day you get a diagnosis, things like that, that nobody on earth, unless you've been through it, will understand. I can try and explain it to you. So that's my second hat. My third hat is involved in the poetry that was mentioned before. I got roped into this via somebody I knew from Swan. It was organised by Brighton and Sussex Medical School and Wellcome Trust, and they did a project called Epigen, which is basically all about trying to get patient experience from the patients in an area that they absolutely don't understand but are feeling completely into a sphere that's translatable to you lot who deal with all of the technology and the wizardry, so that you understand what our experience is in a way. It's all very easy to look at a screen of data or to look at a set of numbers or to look at a graph, but it doesn't really mean anything. And the bit of our experience that we wanted to get across to you lot was what it feels like on this side of it. And that might sound a bit airy-fairy to people who are particularly sciencey, but it really isn't. There's a load of stuff that we need to get across to you that just like one nugget of it might be vital to you guys understanding what's going on. I've overrun time. Can I still read? Okay. So I was gonna. It's easier to read it than it is to explain it. So this is one of my poems from the Helix of Love book. One of the Epigen projects. We collected a load of parents with kids with rare conditions together, and they threw us in a Zoom group with a experienced professional poet, and she gave us some tools. She gave us a toolbox to try and explore some of the things that we felt. This was what came out of it. It's available on Brighton Sussex Medical School's own website, so you can go on there and find this. It's free, because we want you lot to read it. Okay, this is one of the ones that I wrote. It's called Unpacking. I saw one once. Difficult to say I held it since it was under a microscope lens. A nucleus. The tiny dot in a cell. The full stop at the end of a sentence 3 billion letters long. A biological zip file describing a human. We've spent her life unpacking it. Then come the experts. Detectives decoding what this Rosetta Stone might say about what's wrong with our Cleopatra and how it's shortening her muscles, her life. Genius though all this is, do these hieroglyphics tell us about her bawdy laugh and a smile to fall in love with? Thank you very much.
Thanks so much, Paul. I'm actually going to invite everyone to come up on stage, and if you just can take a seat. And I'm going to take moderator's license to start off with some quite broad questions. I want to take us through a few things. I'm going to start actually with where we've just been, which is why does all of this matter? And then we'll move on to kind of how you make some of this real in terms of adoption and wider system issues. But I just want to start off with you, Paul. So, I mean, thank you so much for reading that beautiful poem. I think it would be really powerful for us to hear a bit about the challenges your daughter and your whole family have faced during that diagnostic odyssey. So it'd be helpful to understand kind of the timeline and what you've gone through in order to kind of get to this point.
Yeah, sure. I mean, I think the one thing to say, she was born in 2008, so her journey through all of this, her diagnostic odyssey, has been, you know, an odyssey while everyone was building the path. And we were, I'm pretty sure that Prof. Emma's been across our data on more than one occasion, although she couldn't tell us. But you know, hers would be the centre where all the data would go to. So we went through any number of assays and panels and other things that we didn't understand. And circumstances where we did understand them, you know, we'd get told in an appointment, right, okay, we're looking for this condition. Don't Google it. And obviously the very first thing you're going to do when you get out the door is you're going to Google it. And then you realise the reason that they told you not to Google it is that, you know, you're possibly being diagnosed with something that's terrifying. And, you know, who knew there'd be so many different terrifyings? Who knew? I guess the odyssey has been for us really long and complicated and mad. She was born in 2008. We went all the way through DDD when that came up because we saw that as it was absolutely a DDD, squarely a DDD kind of condition that she's got. So we thought, oh, we could come up trumps here. And we went through DDD. We took samples for that. That didn't turn anything up. So we had to go through all the kind of huge hope and then kind of, oh, there's no finding. And then we found out about the 100,000 Genomes project. And again, we revved ourselves up again. And luckily the 100,000 Genomes project, we were in the lucky third. The 100,000 Genomes project turned out a result that said we've got an error in CTBP1. Never let geneticists name anything. I think you should have a naming department. The condition that produces is called HADS, HADDTS, and that's confused with another syndrome that's also kind of like the same, except it's got fewer D's or more S's or something. Who names this stuff? Find a person, name it after them. The challenges we've had. Two of the things that stuck out to me this morning about the whole of what goes on in the diagnostic odyssey. I'm looking at Dr. Danny's slide this morning about the snakes and ladders scenario. That absolutely struck home. You know, that is exactly what parents go through, except every time you go up a ladder, it's like with an extreme amount of hope. And every time you come down the slide, you go on your arse and it hurts. And I think that's the kicker to take away from it, that every single step in that process is painful for a family, you know, and talking about it even, you know, and bringing that up again kind of is painful for a family. So I think that's something to bear in mind.
So then when we talk a lot about, you know, trying to reduce the diagnostic odyssey and trying to get earlier diagnosis. I mean, what would have been different for you if you'd had an earlier diagnosis?
Well, a lot of things would have been different. I guess there's kind of two tracks, I suppose. One would be what would have happened if. So let's do that first. That, you know, if we'd have had a single process where we came and had a long read set of stuff done with the Oxford Nanopore magic, then it would have been, even if we'd have had to wait, 12 months or 18 months or 2 years to get the data back, she'd have still been, I don't know, like 6. And transposing that into the future if we'd have known what's up with her at 6, then because of the condition that she's got, so she's got a condition where it alters her proteins, so her muscles are all wonky. And so that's a baked-in condition, so it doesn't matter what kind of treatment you come up with. It's baked into every single one of her muscles. She's outgrowing her own strength, and that's what's going to finish her off, and there's no two ways around that. That's our future. But if we'd have found out when she was 6, that would have been a different game. And all the parents of all the kids in our little cohort, there's 12 kids in the world with this condition. We all know each other. We all communicate on Simon Searchlight. That's the other thing that's kind of great about the modern world. There aren't many of us. They're 1 in a billion kids, but we all know each other now because of the internet. So some of those other families have got kids that are young, and they can benefit from this massively.
I mean, it's just really powerful stuff. I mean, it reminds, I think, all of us why this matters, why we're trying to do any of this, and the impact that potentially make. And I know that a number of the clinicians on the panel will have experienced that from another perspective, from seeing what that feels like to communicate with a patient and their family. But in many cases, you're not able to get diagnosis. In some cases, obviously, you are. But especially in cases where, so at the point where you are able to make a diagnosis, what does that, you mentioned about potential for treatment. What does that also mean for the family in terms of what your day-to-day feels like? What tests you're going through?
What do the tests feel like, or what does the potential for a treatment feel like?
No, so potential for treatment you've talked about already, but I'm particularly thinking about the number of tests which you're going through.
Roger.
So I know you've talked to me about that before.
Yeah, I mean the problem with having an undiagnosed rare condition is that if it's a peculiar one, then you can be put through all kinds of mad diagnostic tests. I mean, the one thing that we've got in our favour is the fact that that Nena, my daughter, is particularly resilient to all this antics. So you can take blood from her till the cows come home. She doesn't bat an eyelid. She's like extremely cheerful about everything. But the one time we had muscle biopsy tests, which weren't all that pleasant. But the one thing that got her was she had to have a nerve conduction test, because obviously it's a neuro condition. So they did a nerve conduction test. She didn't like that all that much, and I tried it out after that because it's the one thing I've ever seen that made her cry in an appointment. So I had to have a go, and bloody hell, she wasn't wrong. But yeah, it's so many tests. To just have to have had all of that reduced down to one test would be that would be just life changing. And multiply that by 2,500 families in Swan. Multiply that by each country that's got it. Multiply that by all the kids and families in Simon Searchlight. You're talking about thousands and thousands of lives you're changing.
When we think more broadly about sort of what it takes to do that, I mean, we're talking about replacing, in many cases, a series of tests with something earlier and potentially more comprehensive. But that is no small task. Even with the even once you need to optimize the the data and validate and all of those things. But actually, there's a lot of behaviour which you need to change within a clinical pathway. So perhaps I can ask Danny, Emma, Joris to speak a little to that. So some of the challenges which you see in clinical adoption. So Joris, perhaps I can come to you first, because you've sort of gone through that journey around NIPT. So what can we learn from that around what helps to overcome some of the resistance to change?
Yeah, maybe I'll just jump in the rare disease space, just for people to understand. I think most of us are piloting this in an academic environment. And the reason this is an academic environment is because the tools are not there yet to analyze it. And the talks we hear are possible because we've invested research time. Just to understand, the last 5 years, we started mapping the methylation first to megalodon. We needed to use the Flemish supercomputer, which is really huge. It took 1 week to do one analysis. And this machine brings 24 samples a day. And we needed 1 week, the whole Flemish tier 2, not the tier 1, supercomputer. I mean, luckily then there came a new program, Dorado, and that went a little bit faster. But we're now having a special access to the tier 1. And of course, Flanders, Belgium is a small country, I know, and probably in the UK and the US you have bigger supercomputers. But that's it. We can't just not handle more. So that's the last 2 years. Now we have these tools ready. We have the pipelines streamlined. NVIDIA is throwing out new chips, and it's getting better and better and faster and faster. And I think we're just at the edge of having the tools to really clinically implement the full power of the promise of long-read sequencing. And I think that's definitely what has hampered. And so now it's time for stabilizing some of those tools. We heard some talks where you integrate the one-stop is all, the long-read. But it's like you mentioned, Emma. It's not stable yet. We're still not there yet. However, I think we're there at the moment to implement it. It's not perfect. We still have another 5 years to make things perfect. But if you look in the past, I was very instrumental. I think we were also one of the first labs implementing arrays, one of 10 labs in the world. And we just made choices. Or even if maybe the audience is too young, but if you look at karyotyping, it's not perfect, but we did it, and you say, okay, we missed things. And here I think we're at a stage where we gain a lot. We're still missing things, but it's ready to go. And so that's one of the big challenges in order to allow the clinical adoption. In terms of speed, it might go the same way as NIPT. NIPT was maybe the first genomic test which was very rapidly adopted from the observation that non-invasive prenatal screening was possible based on liquid biopsy DNA and liquid biopsy in the DNA in blood. And the rapid adoption was a consequence of the fact that prenatal screening existed already in another form, was actually very poor. And maybe we have the same paradigm here. We have already exome sequencing, targeted panels, and so forth for rare disease detection, but we know that they're imperfect. And now we have a tool which is better, more comprehensive. Probably, I predict a rapid adoption as long as we have the tool stabilized and maybe a little bit more. That's my token on this.
So perhaps I can come to you on this one as well, Emma, because you've obviously gone through, the journey of scaling towards kind of establishing national clinical genomic services and so on as well. How do you balance the things that Joris has just talked about? So starting small or starting with something which maybe isn't completely perfect but can solve some problems versus taking some time to go kind of big.
But I think that's what we've done with all of these new technologies, isn't it? I guess when you first look at long-read sequencing, and just like Joris said, what you can do in a research setting is really very impressive because you can have one person looking at that data. And that's sort of what Danny was saying. We can do it like that. But when you're thinking about implementing things into healthcare, stability, like Joris said, is absolutely important. It's got to be safe. What you do has got to be safe. It's got to be reproducible. You can't miss something. And we've all the time, as we've introduced new things, we've done it bit by bit. And I guess that's sort of what I was trying to say. We are ready to go with some elements of long-read sequencing where it's providing added value on top of what we already have. And as time goes on, we'll look to replace other functions of what we have with long-read sequencing. And that will enable us to already realise some of those benefits. And then we don't actually have to deal with what you're talking about, in fact, because we've already got we've already won the battle of changing the way that samples flow and things in the healthcare system, at least in England. We've won that battle. We introduced genome sequencing. In a rapid setting, we've introduced genome sequencing across different parts of healthcare. It's disrupted those pathways. It's imperfect in places. But in the rapid setting, actually, it so profoundly benefits patients that clinicians come. We have education. We run education sessions for clinicians online. We used to do it once a month. We do it now once a quarter. We get 200 plus paediatricians from all different parts of our healthcare system who deal with children who are acutely unwell or who use that service. They turn up, they present cases that they can see have benefited from that, and we do two-directional education. So the scientists talk and the clinicians talk, and we talk about those cases and what we can learn from it, and then we have a little bit of an educational session, and people are free to ask whatever question, because no question is a silly question. And that has enabled us to really get people excited about it, to learn from it, for us to learn. We've evolved the service, what patients we take from doing that. And we now use long-read sequencing examples in that education so that people can see that we can get that other bit of value now. All this time that Emma and the scientists have been saying to me, if we've only got one parent available, we can't offer the same sort of test. Well, now we can do some things that they said we couldn't do last year. So we now would be thinking slightly differently about that. So we get round it by bringing everybody with you. We have a regional lead for rapid sequencing in all the hospitals in the UK from clinical genetics or medical genetics if you're from the US. And they help us to make the service work. They turn up to those education sessions. You probably tell I'm quite passionate about nationalised health services, but I do think that it really works, actually, when you are trying to introduce new things. You have huge amounts of data to inform the decisions that you make. You have access to outcomes. It's why Genomics England has been successful, why the 100,000 Genomes Project was successful.
So I've just realised that I'm the Atlantic over here.
Yeah.
And so perhaps we can cross over.
Yeah, cross over to the Atlantic.
To Danny, because actually we were talking a little bit about the kind of some of the structural differences in health systems, but you obviously do some similar things, albeit in a very different payer landscape, and the healthcare system is structured in a very different way. But can you speak a little bit to how you have engaged clinical colleagues around different approaches to innovation, particularly in N4 sequencing?
Yeah, one thing I'm struck by as I listen to the first part of the panel is, has any technology so quickly entered the clinical space? It once we see that it's actually there's utility, all of a sudden everybody wants to use it, right? Your colleagues from pathology, from infectious disease, from the neonatology service, they all hear, oh, you can do this. Can I try to solve this new problem or this existing problem, this old problem, using this new technology? I think it's been really interesting to be a part of that. And I think that's really kind of driving that uptake, that interest. And it's, in a way, it's almost overwhelming. I don't know how you guys feel, but there's so much to do, and there's just not enough of us to get it implemented. So it's been fun to be part of it from that perspective, but also a little bit overwhelming. And then we kind of think about, well, we've quickly shown in the research space, there's utility. We can solve unsolved cases. We can solve them quicker. We can get more information. But in our system, how do we transition that into a clinical test? And the problem we immediately run into is the payer problem. How do we get this reimbursed? The individuals in the lab who are doing the testing see the value. They see that we're going to move into this space, but now that we're kind of at the point where, how do we make sure that we're getting reimbursed for this, right? We know that we're probably going to eat that cost for a little while, but how do we really get this to patients as quickly as we can?
So Nick, I feel like you know where I'm coming with this. So particularly, within United Health Group, obviously big payer in the US. So how do you approach, how do you view these kind of challenges Do existing payment mechanisms do the job? How do you make the case for something like this entering clinical service?
Yep. Let me do a sort of across the pond thing as well. So the visceral point is something that I think probably we all one way or another carry with us. So David Cameron was the father of a son, Ivan, who died at the end of a long diagnostic odyssey, and David has the story of like lying on the hospital floor of Great Ormond Street during his campaign for the 2010 election, and sort of sleeping on the floor with Ivan, who had a rare epileptic condition, Ohtahara syndrome. And that there's a direct line of sight between his personal experience and the commitment to the 100,000 genome program, and then to establishing Genomics England and all the momentum that's followed on. And actually, we of course have the same kind of personal stories within the membership of United Healthcare. And the way that we look at it is because the American system is super economic as well as everything else. So people on a diagnostic odyssey cost the system a huge amount of money. There's a huge amount of emotional cost and a huge amount of financial cost. And they absolutely hate the healthcare system and they hate their payers. And so a number of years ago, we set up a program called the Diagnostic Odyssey Program, or the Special Needs Initiative. About 5 years ago, there's no testing that we were proactively pushing in that. That was all about using claims data to identify suspected children that were suspected of being on a diagnostic odyssey, and then creating a wraparound advocacy solution for them so that we could actually give heavy-duty support for the families to figure out a number of different. We send to like Boston Children's, Rady, Mayo, and in the about 2 years ago, United Healthcare made a policy change to cover whole genome sequencing for rare undiagnosed genetic disease in the outpatient setting as well as the inpatient setting. And we're just about to launch a program to test out what are the barriers to actually that happening in the wild. Because as Nabiha knows, over the last I probably shouldn't say this, but over the last period since that policy change was made, less than 100 people have received a whole genome sequencing as a result of that. So there's something about the transmission mechanism that we've got to figure out how to actually drive from not just covering and reimbursing a test to actually ensuring that we actually get utilization of it when it's appropriate. So this program is designed to, at the point at which the patient is suspected of being on a diagnostic odyssey, immediately push them towards either an inpatient or outpatient whole genome sequence, and start to chart what the barriers are to clinician adoption, or whether or not there are all sorts of other things. So we're pretty committed to trying to find not just kind of, I think building the clinical evidence is critical to get payer coverage. It's commercial payers, that's an absolutely fundamental factor. Because as I mentioned earlier, the FDA isn't your gatekeeper as it is with drugs. There's professional societies which take different perspectives. So you have to build up a really strong amount of clinical utility evidence to get to coverage. And at that point, how do you get into guidelines? And then how do you get to promote that through the system? So there are a number of different steps that we're working on, but we're going at it from every angle at the moment.
Although I will say it's quite remarkable hearing some of what we've heard today about some of the routes that people have been taking to, in many ways, circumvent all of those different steps and all of those different barriers by just doing things which are very innovative and demonstrating success. What I will probably do now is to open up for broader questions. I think we've got about 20 minutes, and I know that there will be a ton of questions, both in the room and I know also coming online. So I'm going to ask colleagues if they could, I don't know whether you can switch, there we go. So we've already got 7 questions. Oh, Paul, please show us your shirt. There we go. That's I don't think we've ever been asked for that question at an Annapol conference before, but okay.
There you go.
So what I think I might start with, just because we have just been talking about payment mechanisms, is moving slightly to the second question here. And meanwhile, please be thinking of your questions here too. And if you put your hand up, then we can bring a mic over. I think there's one down here, and there's another one in the middle. Okay, so let's start off, Joris. So there's a question here around biobanking, given your involvement in biobanking in Belgium. So how do you see the future of biobanking contributing to advancements for rare diseases?
Okay. It depends a little bit what we understand about biobanking. I think Dan has alluded to it during his talk. I think if the biobanking is the in silico variant databases, they will bring a humongous amount of, make interpretation of genome information much easier. We're lacking the big databases for structural variation, but even the SNV databases, I think population-specific SNV databases will improve and speed up the diagnosis. Just to give an example, I mean, I'm living in Belgium. I think most of you know that the Netherlands is just flanking us. And there's a genome of the Netherlands database. So we compared what's the added value of having a database of our own. And actually 5% of rare variants are actually not captured in the database of the Netherlands, but just creating our own. So I think there's a huge gain to be made in variation databasing. And I think especially the telomere to telomere consortium, who has now mapped those unmappable regions, I think that's just a black hole at the moment, and these need to be filled with reference databases. So I think that's one thing. If the biobanking means, do we need also biomaterial? Well, we heard this morning, or we know we have 40 or maybe 50% yield at the moment of all the patients. I think long read will add somewhat, but we're not going to get to 100%. So cell lines would definitely help in the future to do functional assays, look at transcriptomes. That's obviously one thing. And these rare diseases are extremely rare. So what we really need, and I'm actually working on some tools to do that, is know where or which diseases and cell lines available. Have IPSs been made by people or not? Have different tissues been stored or not, and can we get access to that? And I think the problem is that everyone has his own register, but we don't know from one another. So just my group has been trying to make some federated model where we would allow access or not necessarily access, but knowing where's what so we can reach each other. One thing is to do that centralized, but I think that's maybe not doable. But I think there's room to bring things together, and that would tremendously speed up rare disease diagnostics, I think. Anyway, just
so huge opportunities there through collaboration. And we have, I think, a couple of questions on the floor. So at the front here, and then come to you, Stephen.
Hi, that's a great discussion. Thanks a lot. As a researcher, I really wonder, how does that look like on a daily basis in a clinic? So let's say you have a patient, you find a cause, that's a new random gene that you identified. How does that translate to what you have available to treat the patient? And how do you see that changing in the future?
I'm going to come to Emma first and then Danny.
So it depends in what setting. So I guess we still have to be honest that in the majority of cases, what we provide for people is a bit like what Paul said. We provide an answer. We don't necessarily provide a treatment, but an answer cannot be underrated.
No.
An answer is really, really, really important to people. That's why organisations like SWAN, Syndrome Without a Name, exist. It allows people to connect with each other. And even if you've got a child who is essentially an adult in age terms, you still want to know what their future holds. In fact, you probably even more want to know at that point in some senses, because you want to know how you're going to look after that child when you need looking after yourself in the future. And so having some idea about is there a kidney problem? Is there another problem that my child might get or I might get is really important to people. So absolutely, my hope, like Danny was talking about, is that we'll be at in that situation where you get an answer for a patient straight away and you've got a therapy. But providing an answer for people can give them hope for the future. It can allow them to plan their future, and in some cases, it can be that you're considering withdrawing care on a neonatal unit, and you can go from essentially sticking things in that baby up until the moment that they pass away to saying, okay, this child has got a really devastating disorder, but we can transfer them to hospice care and they can die what I would term a good death with their parents with them in a planned way. So I come and I talk about where we've saved children's lives or changed their lives with rapid sequencing, and I hope more and more that's where we will be. But we've probably changed as many families, if not more families' lives by giving them some sort of information. And the saddest times for me are when I have to say to families, I only know that there's 2 other children in the world with this condition. So we've got an answer for you, but we know so very little, and your children will help us to learn for other people's children. And even that can mean things to families, can't it?
Yeah, can I pick?
Yeah, of course.
I mean, that's a huge thing. It's a huge thing. And also, I guess in that situation, I think it's important to say to all the clinicians in the room that never underestimate the ingenuity and the drive of families under those circumstances. So you talked about, fictional condition B, where there's 2 kids in the world. You can bet your bottom dollar within a 12-month, that one family that you've met in Exeter will then know the other family in wherever. Even if there's language barriers in the way, the families will drive towards those things.
100%. I've just come back from a family meeting in Maryland for a disorder we described in 2015 in the Amish community. And now there was a family who started an organisation called the KPTN Alliance. And in that family meeting, we've got families that have come from everywhere. Yeah. It's a huge growing group. And they've driven the research. They're the ones standing behind me with a stick going, apply for that NIH grant, please. Have you contacted clinician XYZ? They're the ones pushing us. And you're absolutely right. People get, people are motivated to get the answers for themselves, aren't they? And to push behind us, pushing us forward.
I mean, I think the day that you get a diagnosis, the day you kind of step through that door into knowing rather than not knowing, it's brilliant and awful all at the same time. It's a very difficult thing to describe. And ultimately, it boils down to one thing. If you've got a diagnosis, you're either going to be in what I call unimaginable scenario A or unimaginable scenario B. Unimaginable scenario A is that you outlive your child who's got a rare condition and you're not going to be there to look after them. Unimaginable scenario B is that they don't, and you don't get to see them grow up. But having a diagnosis, and even if it's a diagnosis for something that's vanishingly rare, and you're cobbling together your own kind of guesses from cohorts from scientific papers that you've got to learn how to read, even under those circumstances, that gives you more information than you had before, and that's always better than nothing. I could ask any single one of the families in Swan UK, and all of them would rather know than not. All of them would rather know. So that very simple, fact of giving us a solution, even if it's a solution we don't understand, or even if it's a solution that nobody understands, that's still better than no solution at all. Always is.
Can I just ask Danny, so from a therapeutics perspective, there are sadly too many cases where we're not able to treat, but increasingly there are some where, albeit with highly tailored therapies, you are. Could you perhaps speak a little bit to that and to the kind of the process which goes on?
It takes too long today, but the goal is that we speed it up to happening within days to hours even. I'll just say that I've been involved, I think, on a research basis in kind of the gamut of cases where, you have an unsolved condition, you do a research study, you identify a variant. It's often a challenging variant, an intronic variant, a structural variant, and then the fact that it's challenging can open up unique treatment opportunities. So things like antisense oligonucleotides, where you may be able to alter splicing, you may be able to tamp down expression or drive up expression, and you really need all the information that you can get with long reads. Phasing the entire gene, with all the variants on each haplotype, because often you may not be targeting the pathogenic variant, you may be targeting a nearby variant. So having long reads gives you, it kind of makes that process go a lot quicker, has been our experience so far.
I know there was another question in the middle of the room. Yes, there we go.
I don't know if we can hear it from, if we don't.
We will try.
So, great discussion. I did have a question about strategy. And I think that most of the people in this room would, at least some of us would agree with Danny's comment about long-read genome sequencing could be the one test that rules them all and replaces many tests. But that may be compelling to us as geneticists, but what arguments are the best arguments we can make to laboratory directors, to clinicians, to patients, to the public, policymakers, and payers? And I'm specifically thinking about one possible argument might be it increases efficiency because it's a single test. Would it also be a cost-effective solution? And the third thing, which I haven't heard much about, is it's a better test, and the diagnostic yields will go up. There's been a lot of anecdotal presentations and posters and talks over the last couple of days of individual examples where long read was the key to getting a diagnosis. But how often does that happen? Any guesstimates?
Okay, so I'm going to go to Nick first for like the broader perspective, and then I probably work my way around, because I think this one there is value in going pretty much across the panel and giving some of the perspectives here.
It's a super question. The challenge I think I mentioned in my opening comments was, how do you integrate cutting-edge technology into the healthcare system? And very often what I see is test developers, diagnostics developers, talking to the US payers like the Englishman abroad. When not first understood, raising your voice slightly louder and getting louder and louder. My test is the best thing in the universe, and showing another glossy marketing deck with 45 pages of really cool, like, glossy marketing. The currency that US payers deal in is clinical utility. And so for the tests that have been successful in the marketplace, the NAPTs, the Oncotype DXs, the Cologuards, increasingly looking like comprehensive genomic profiling, there is a very strong correlation between evidence, publications, coverage, and adoption. And so I really think the most important job is to put together the data that shows the clinical utility. Now, that's not straightforward. And again, what the payers are interested in is, from the validity point is, does it work? And the utility point is, does it measurably improve outcomes? And those are not straightforward things to demonstrate. And you need to construct the studies in the right way so that the endpoints are going to work. So that that can be very expensive, obviously, to do those prospective studies. But that's ultimately what's necessary in order to persuade at least the US payers on the commercial side to cover actually any technology. So superiority in terms of quality, reduction in terms of cost, ideally a combination of both of those. But those are the two key things that you need to be able to show.
Danny?
It's a fantastic question, and there's so many pieces to it. I want to address the lab directors and the laboratory piece. I think it's getting harder and harder in the U.S. at least. I can't speak for other parts of the world, but hopefully others can. To staff your lab, to maintain your competency, to keep reagents on hand, even, right? So any piece of that you can simplify, I think is an argument for. If you can simplify the whole process, I think that's an argument for using a single test. You can simplify your computational workload, do things with less individuals, and really kind of drive up your diagnostic rate. Although you're right, I actually am trying to talk less about diagnostic rate. I think I'll pick that back up in a couple of years after we get these tests implemented. But for now, let's just make sure we can show that these are not inferior tests, and that we are doing the same. We're able to get the same information. also just want to comment really quickly on we're going to lose a little bit of sexiness with long-read sequencing. And what I mean by that is, we're often trained as geneticists to say, I think this is condition X, so I'm going to order test Y that specifically diagnoses that, right? And I think that mindset needs to change. And my personal example is a few months ago, I saw a kiddo who looked like she had Treacher-Collins syndrome. And I sent an exome because the family was from Alaska. And if I was wrong, I didn't want to make them come back to be reevaluated and to, get another round of testing. So I didn't test TCOF1, I just did the exome. They didn't have United Healthcare, don't worry. But I just, you have to be okay with that for things even like achondroplasia, right? That this really broad test is going to pick up some very straightforward, simple things, but that's the system that we will be working in someday. So.
So I'm going to come to the side.
There were indeed different elements. Maybe I'll address the question of where are we with the clinical utility, because we need to prove the added value, and I think to do that it's not trivial. Actually, I'm still running a clinical utility or a multi-centric trial, prospective trial in Belgium, just with short read, where we're doing parallel standard of care whole genome sequencing. And the added value just in the short read is not clear yet, because you're looking at the non-coding elements. We don't have the data yet. So there's this suspicion that we will increase the diagnostic yield, but we need the data, and it's circular. So we need data on the whole genome. We need this variant database in order to be able to interpret the non-coding genome. And then probably we will be able to start mining those, but that's circular. So at this stage, we're limited to what we know. And what we know is from the short reads. So the added diagnostic value will be, I don't know marginal, but will be incremental. And it will not be solving the other 50%. Maybe it will add another 5% maybe, but we need to prove that. And so we need large-scale studies. And of course, who can pay for such large-scale studies? No, it's not trivial, and I think we need to prove that. I think indeed we have now smaller-scale studies showing non-inferiority, both in the labs, all of our labs. If I look around, or what I heard on this meeting, I think Genome England is maybe doing the largest scale study, and then this type of information will really give that data. But still, it's circular, because we need to gather data on reference genomes in order to mine the non-coding and the regions we don't know.
So I'm probably going to get told off because we're slightly over time. So I know we need to wrap up, but I do just want to actually come to Paul for one last thing, because I think there was an element there of the question, which is about how does this need to be effectively communicated to patients and their families? So some of the uncertainty which comes with this, but there's often questions about how you deal with variants of unknown significance and so on. From your perspective, how do you deal with uncertainty? How do we effectively communicate that with patients and their families?
Regarding the variants of unknown significance, I think that's a tricky one, because we've never been through that. We've never been through that experience. So I suppose you've got the wrong patient example in the chair, really. I mean, I suppose the uncertainty piece is definitely, up until the point that we got a diagnosis, every single day was uncertain. The uncertainty is what you live with an undiagnosed condition.
Well, if you think about it from when you were talking about it, when you were saying somebody said to you, this is what it might be, and you're waiting for that test.
Yeah, those moments are pretty terrifying, I think, especially when you know it's going to be something that's really life limiting or really debilitating. I think those moments are they're awful, but from the point of view of a practitioner, I suppose, so long as you're empathetic about it, then that's your job done. I think there's nothing else you can do to sugarcoat those pieces of knowledge, I think. And again, I think not being shy to tell it how it is. You think, if you're delivering diagnosis of of something like that, I think it's quite difficult. And the families will go away and make that they've got to come to terms with that themselves. I think there's no other way around that. I don't think.
I think sadly, yeah.
And I just ask, I think the rules are that we do not report variants of unknown significance. I think it's toxic information. But maybe
that's our way of. We would only report an uncertain variant if we had something we could do to change the uncertainty.
Yeah.
And I think that's the safe way to be.
Yeah.
And unfortunately, while I think we could carry on this discussion for another hour and a quarter for sure, if not another entire London calling. But just thank you very much to all the panellists. I'm really sorry we've run out of time. But please join me in thanking everyone for such a wonderful discussion.